Shiv Prasad, can you tell me what I took in the last class? You were absent, I guess. You were not there in the class last week. I know you were not there. Yes, Shiv Prasad, can you talk? If you don't talk, I consider you're not there.
Ma'am, we
Yeah.
discussed about gram positive and gram negative bacteria, ma'am.
Okay. You were there last week
How we,
when I took the class.
ma'am?
You were
I
there
have
last week.
attended, ma'am.
Last week you were there when I took the class.
Yes, ma'am. Yes, ma'am.
Okay. And Manikanta, you were also there, so you also know. Hmm?
Yes,
Or
madam,
kaun
I
tha
am
last
also
time?
absent.
Yeah, there were two Akhilas last time, no? Akhila Patel and one more Akhila. Are you there, Akhila, today? Akhila, are you there today? Akhila Patel is there. You're you're still getting inside the class, so I need to wait for 5 minutes to start, or shall I start? You have any other class today after this? After 2:30, you have any other class?
No, ma'am.
No? Okay. So you are getting used to this online timetable, huh? Okay. Shall I start or anybody else is going to come now? Shall we start with the class? Can you see the screen? Can you all view this?
Yes, ma'am.
Okay. I'll just go through the slides for those who are absent. Anybody who has come today and they were not there in the last class? Is there anybody like that?
No, ma'am.
No? All of you were there in the last class?
Yes,
Anyone who
ma'am.
has joined today?
Yes, ma'am.
So all of you know, I'll just, or you want me to brush up through these slides? All of you were present in the last class. You remember
Yes,
all
ma'am.
this? We've done all this prokaryotic cell, eukaryotic cell, okay? The
Yes,
structure
ma'am.
of prokaryotic cell, the different organelles in prokaryotic cells, eukaryotic cell. I'm just brushing up through everything because we've already done in this in the last class. We'll just go through these slides. Cytoplasm, we were discussing about various parts of the prokaryotic cell, okay? Then this is the ribosomes. Then we discuss the plasma membrane. You remember discussing the plasma membrane? This fluid mosaic structure, we have discussed about this, which has hydrophobic and hydrophilic ends. Do you all remember all this?
Yes, ma'am.
Did anybody go through the notes which sir has given, the PDF which sir has given? Sir has sent a complete PDF of your unit. Did anybody go through this? Did you all open the PDF which sir has sent? No? Nobody has opened the PDF sent by sir?
No, ma'am. I saw it, ma'am.
Hafsa,
But
We have opened, ma'am.
But actually, because it is new for me, but I don't know. I little translate it.
okay. That is Hafsa, no?
Yes, ma'am.
Yeah. Others, anybody, did you see that PDF sent by sir?
Yes, ma'am.
Who is that?
Gautam, ma'am.
Gautam. Okay. So, did you all see the topics? Did you all go through the topics? Nobody has yet seen, I think. Just open and go through the topics also, okay? Okay, so bacterial cell wall, peptidoglycan layer, we have done that day. It has NAG and NAM. Okay, NAG is what? What is NAG? N-acetyl glucosamine, then N-acetyl muramic acid is NAM.
Muramic acid.
Okay, these strands are connected by interlocking peptide bridges. Okay, we can see the image over here, the peptide bridges over here. This is the oligopeptide bridges which connect the NAG and NAM. So remember this, don't forget N-acetyl glucosamine and N-acetyl muramic acid, okay? So this also cross section of the cell wall we have studied the previous class. This is the positive cell wall. I've explained you the difference between the gram positive cell wall and gram negative cell wall. Okay. See, this peptidoglycan layer is a very thick peptidoglycan layer in the gram positive cell wall. Okay. Whereas here you can see the layer is very thin. And then there is teichoic acid also present in gram positive cell wall. Can you see this? Teichoic acid present in gram positive cell wall. This is again a structure showing you the cell wall of gram positive and gram negative. Okay.
Sorry, ma'am.
Ma'am, I'm
Yeah.
unable to see screen.
Me too,
Same,
ma'am.
ma'am.
Me too.
Yes, ma'am.
Array, I asked you all before starting, no? Nobody is able to see?
No,
Yes,
ma'am.
ma'am.
Share
Yes,
the
ma'am.
screen, madam.
Array, tu batana na when I started.
We are not able to see the screen, ma'am.
I asked you all, can you see the screen? You said yes. Muzammil, kindly tell me whenever if you all are not able to see.
Okay, ma'am.
Okay. I asked you, no, before starting. Someone said yes, we can see. When I start, you should tell me, no. How will I know? Is it okay now? You can see.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
I asked you
Okay.
5 minutes back also. You all said you can see. When I opened prokaryotic cell structure, I asked you all, can you all see? You said yes, I can see. Who said that? Yes, we can see. Now is it clear?
Ma'am, we thought that you asked about your screen, ma'am.
I'm asking about this, whatever PPT I'm showing you, I'm asking that. Because if you all don't respond, how will I know? See, I went off 10 slides, 10, 15 slides, I went ahead. And then you all are telling me you're not able to see. You should tell me, no, right at the first slide. How will I know you all can see or not? I thought it's already in screen share, is already going on, I thought. Okay. Again, I'll say all this for you all. I was showing this to you. Did you see this? Abhi dikh raha hai?
Yes, ma'am.
Yes, ma'am.
Yes,
हाँ,
ma'am.
तो तब क्यों नहीं बताया नहीं दिख रहा करके?
Yes, ma'am.
Okay. Now again I'll talk about all this. Okay. I'll just go through the slides. We have done all this. Okay. Cytoplasm, granules, prokaryotic cell. All this we have done in the last class. I'm just showing you so that you get a recap of everything, because you tend to forget, no? Because you all are having so many classes in between. So you tend to forget what I was taking and what we were doing. So this is just a recap for you all. Remember all this? All of you? I hope you all remember this.
Yes, ma'am.
Plasma membrane barrier we have done gram positive and gram negative cell wall. I told you about the staining, why this comes pink color, why this becomes violet color. I explained all this to you all in the last class. Peptidoglycan cell wall.
Yes,
I
ma'am. Yes,
explained
ma'am.
to you all. See, you need to respond continuously, because I will not know you all are there, you all are understanding or not. That's what I'm telling you. When I'm talking to you, respond. Okay. So this is what I was showing and explaining to you all. I was assuming that you are able to see this. Okay, this NAG and NAM, these strands, N-acetyl glucosamine, N-acetyl muramic acid, and the peptidoglycan.
Muramic acid.
Yeah, N-acetyl muramic acid and acetyl glucosamine. Okay, they are linked by the peptide bridges. These are the peptide bridges which they are linked by. So this is the thick peptidoglycan layer which is present in the gram positive cell wall. That is what I told you, the primary stain which we add, the crystal violet. Primary means the first stain which is added. So the primary stain which we add, that is crystal violet, that penetrates into this peptidoglycan layer, and then it doesn't go after washing or rinsing with alcohol. Okay. This is what I had explained to you in the last class. I hope you all remember.
Yes, ma'am.
Yes, ma'am.
Yeah.
Yes, ma'am.
Okay, thank you. This structure, okay, see this peptidoglycan layer, you can see this thick peptidoglycan layer in gram positive, and whereas it's very thin in gram negative. Okay, so this is, and this is a cross section view of the cell wall, right? You can see this gram positive cell wall and gram negative cell wall, and the thick peptidoglycan layer. This is a thick peptidoglycan layer which makes the thick wall very rigid and thick. Okay, that is the reason this takes the crystal violet, and this gram negative, after washing, what happens? The gram crystal violet color gets decolorized and saffronin penetrates into the cell wall, showing it as pink or reddish pink in color after staining. In this slide we saw, no, ये देखो ये ग्राम नेगेटिव कैसे दिखता है, पिंक और रेडिश पिंक, and this is violet color. This will be, ये जब माइक्रोस्कोप में तुम देखते हो, it will be this color. You can see my cursor, no? You can see the
Yes, ma'am.
cursor. Yeah.
Yes,
So
ma'am.
this will be actually this color, this violet color. You can see when it is staining. You'll be doing staining once your college starts. You'll be doing staining, gram staining and all. I explained gram staining in the previous class. I hope you'll remember what is gram staining.
Yes,
why
ma'am.
why we do gram staining. All these slides I've explained to you with different cell structures, gram positive and gram negative cell wall. And we were discussing there are two additional layers of the cell. For prokaryotic cell, they have two additional layers which can be, they're called as glycocalyx. We discussed this in the previous class, glycocalyx. And the two additional layers, it can be, it is called a slime layer, and one can have a capsule also. The cells can also have capsule. I told you slime layer is what? It is a glycoprotein loosely associated with cell wall. We discussed this, you remember? Slime layer
Yes,
causes
ma'am.
bacteria to adhere to solid surfaces and helps prevent the cell from drying out. The slime layer of gram-positive Streptococcus mutans allows it to accumulate on tooth enamel, mouth, and one of
Yes,
the
ma'am.
causes of cavities. Okay, so slimy, the word slime itself says that it is smooth slime layer. So what does this layer do? It attracts the other bacteria, and the other bacteria also accumulate on this, resulting into cavities. The next one, which we additional layer, is a capsule. So few bacteria we were discussing. See, this is the cell, the prokaryotic cell, and some of them have a capsule layer, capsule coating over it. Okay, that is polysaccharides firmly attached to the cell wall. Capsules are there to solid surface and to nutrients in the environment. Capsule also protect bacteria from being phagocytized by cells of the host immune system. So it is a protective covering for the bacteria. You can see this bacterial cell inside, this red one, and here this is the protective covering around it, which protects the bacteria under tough conditions. Okay, and it protects from other bacteria, or by getting phagocytized also. From phagocytosis also, the capsule protects the bacteria from phagocytosis also. Okay, and one of the next thing is endospores. We need to study about endospores. You all have heard about endospores, I guess. In 12th we study about endospore also, you know?
Yes, ma'am.
Yes.
Yes,
So
ma'am.
what are endospores? What are endospores?
Reproductive structures.
Non-reproductive structures,
Non-reproductive structure.
small number of bacteria. Okay, so what is that? It is again a coating. Okay, it is a spore which is produced by a few bacteria and are commonly found in soil and water. So endospore is also a covering. See, this is called as endospore staining. You will be doing this also you have in your syllabus, endospore staining. So see, the coating, the bacteria is red and the spore is green in color. These green colored are the spores. So what happened? Endospores stain bacteria were of Bacillus subtilis, showing endospores as green and vegetative cells as red. Okay, so this spore takes the green color. Okay, this malachite green is the stain which we use for it, and after staining, this malachite green penetrates, we heat it. Okay, in normal gram staining we don't heat it. We were discussing gram staining in the last class, you remember? So
Yes, ma'am.
we had crystal violet, then we had alcohol, then iodine, and we have saffron and also in the protocol. So there is no heating in that protocol. But in when you're doing endospore staining, we need to heat it. Okay, why we heat it? We heat after we add the stain and we heat it so that the stain penetrates the cell wall. Okay, so that the stain there in gram staining we were only staining the cell wall. You know, gram staining में क्या कर रहे थे? We were staining the cell wall. The cell wall was taking the color and showing us violet or pink color. But in endospore staining, what we do? We want to see the spores. ये जो green color के हैं, ये we want to see the spores inside the cell. So we are trying to stain the spores which are present inside the cell. So what are we doing for that? We have to do heating for that. Are you understanding, all of you? Akhila?
Yes, ma'am.
Yes, ma'am.
Akhila?
Yes, ma'am.
Yes, ma'am.
All of you, is
Yes,
it
ma'am.
clear what I'm trying to tell? So in gram staining, we only stain the cell wall of the bacteria, and from the cell wall we conclude that it is gram positive or it is gram negative. How do we conclude that it is gram positive or gram negative? See, bacteria can we see with naked eye?
No.
No, ma'am.
No. Okay. So to visualize the bacteria, what we have to do?
Staining.
Staining. Who said that?
Muzammil.
Yeah, good. So to visualize the bacteria, we do staining. So staining. Now I told you bacteria. Now you can do it with any stain. You just if it is just observation of the bacterial cell. Okay, if you're just observing the morphology of the cell, or if you just want to observe the bacteria, you can use any stain. But if you are differentiating with gram positive or gram negative, I told you they're classified into two as gram positive and gram negative. Okay, so you are classifying the bacteria between two categories, that is gram positive and gram negative, depending upon their cell wall. Okay, depending upon their cell wall, you're classifying them into two. And so what were you using? You're using two stains, crystal violet and saffronin. I told you the primary is the first one, secondary means the second one. So how are you classifying it? The primary take which the positive take which color?
Crystal violet.
Crystal violet. And the negative take which color?
Saffron.
Saffron.
Yeah. And the negative are you when you see under the microscope, the negative are seen in which color?
Saffron.
Yeah, and they will be seen as pink or
Pink
reddish.
or red.
Pink or reddish.
Yeah, and this is because of the cell wall. Cell wall में क्या है? Peptidoglycan. The peptidoglycan layer is very thick and rigid in positive, so it retains the primary stain. Okay, but now that was about a cell wall staining. That was cell wall staining, gram staining. Now here endospore staining is you're trying to stain the spore which is present inside the cell. Okay, some bacillus like Clostridium, they have spores. Okay, this spore we have to we are trying to penetrate the stain inside the spore. So how? So because the spore is rigid, okay, because the cell endospore, the cell wall will be very rigid. So for the stain to enter the cell, we have to heat it. Heating में what happens? The pores open, the cell wall pores open up and the stain penetrates inside the cell wall. So this is under microscope. When you see, this is the view under microscope, it will be red, and in between you can see green spots. Okay, you will see a red long cell and in between you can see green spot. So that will be how you'll be spotting the spores. Okay, and this is a diagram showing this. You understood what endospores? You understand, all of you? What
Yes, ma'am.
endospores? Yeah,
Yes, ma'am.
they have, and these spores, they have resistance to radiation, desiccation, temperature, starvation, and chemical, and also tough environmental conditions. They're capable of surviving in tough environmental conditions. Okay. Then we're talking about gram positive obligate or facultative anaerobes. They are also classified into aerobic and non-aerobic, anaerobic. Okay, I1 is the classification which classifies them into gram positive and gram negative depending upon the cell wall. Okay, then they're further they are also classified into aerobic and anaerobic group. We understand what is aerobic and anaerobic, no? What is aerobic and what is anaerobic?
Aerobic is in the presence of
Aerobic.
oxygen Anaerobic is in the
Anaerobic is
absence
the
of
absence of oxygen.
So the cells, aerobic means the cells require oxygen for their growth. And here the anaerobic means those cells do not require oxygen for their growth. So that is how we classify them. So they will grow under aerobic condition and few will grow under anaerobic conditions. Okay, that is how we classify them. So one more classification is aerobic and anaerobic, and there is a group called obligate or facultative anaerobes. Okay, so they are those which can survive in both the conditions. Okay, facultative and obligate, when we see, they are those which can survive in both the conditions due to extreme tolerance to both heat and used to test heat sterilization techniques and chemical disinfectants. Okay, two groups of bacteria produce endospores. Which are they? Two groups of bacteria which produce endospores. See, this is endospore staining, this is gram staining, and this is a bacillus. Endospore produces bacillus shape. The shape will be bacillus. You have done shapes of bacteria. You have
yes,
any
ma'am.
idea about shapes of bacteria?
Yes, ma'am.
Yes, ma'am.
Okay, that is the next one. We'll do with this appendages and flagella first. You know what are flagellas? What are flagellas?
They
Flagellas,
help. They help in
Small
motility.
hair-like structures, ma'am.
they are useful in motility. Okay, flagellas are useful in motility. They are extension appendages of the cell. Okay, they consist of delicate strands of
Locomotion, madam.
protein. What happened? Anybody said something?
Helps in locomotion.
Yeah, helps in locomotion. Okay, they allow them to move or adhere to a solid surfaces. So these are the strands. You can see these thin strands. They are called as flagella. Okay, flagella again can be monotrichous, peritrichous, lophotrichous. Have you heard about these terms? Monotrichous, peritrichous, lophotrichous?
Yes, ma'am.
Yes,
Yes,
ma'am.
Yeah.
ma'am.
So that is how they're classified depending upon the flagella, how the flagella is present in the bacteria. Like it is, if it is in one side, or if it present in both the poles of the cell, or it is throughout the cell. So they're classified into such that monotrichous, peritrichous, lophotrichous. You've heard this, no?
Yes, ma'am.
Yeah. Okay, and then after flagellae, there comes the pili or fimbriae. There comes the pili or fimbriae. Is anybody trying to enter? Is there anybody in the class who's trying to enter? I can hear it. Okay, let's see. So, fimbriae, most gram-negative bacteria have these short, fine appendages surrounding the cell. Okay, gram-positive bacteria do not have this, and no role in motility. Okay, these fimbriae have no role to play in motility. Okay, and pili are what? Tubes that are longer than fimbriae. Okay, you have pili and you have pili are the tubes which are longer than the fimbriae, and this help in what? In help in transfer of genetic material. Okay, these are the pili. You have done this. You have done this. Do you know this?
Yes, ma'am.
Yes, ma'am. Conjugation.
Conjugation.
Conjugation, yeah, it is conjugation. This joining is called as joining of two bacteria with the pili is called as conjugation, and this is called as conjugation tube. This is called as conjugation tube, and the genetic material passes through this conjugation tube from one bacterial cell to the another. Okay, this is also called as F plasmid. Or the sex plasmid, which transfers the genetic material. Okay, coming to the shapes of different bacteria. Now, one was they classified depending upon the cell wall. Okay, classification depending upon cell wall, which classifies them into gram positive and gram negative two groups. Next was, it was depending upon aerobic and anaerobic, depending upon their oxygen requirement. They classified into aerobic and anaerobic. Then depending upon their shape also they are classified. Okay, they're classified depending upon their shape also, like if they are bacilli, if they are coccus, if they are spiral, or if they are spirulinum or spirochete. See, these are the shapes. You'll have seen these shapes before.
Yes, ma'am.
Yes, ma'am.
Yes. So these are bacilli. Okay, this shape is called as bacilli. Mostly the endospores, they are in bacilli, the spores. Then these are cocci. When it is round, it's cocci. Then it is, this are called as spirali. So in this you have two, spirillum and spirochete. Okay, when it is flexible, it is spirochete and spiral with rigid cell wall. When it a rigid cell wall, they're called as spirillum. Okay, and these are the examples: Cocobacilli, elongated cocal form; filamentous bacillus that occur in long threads; vibrios are short, comma-like. Okay, if it is comma-like, it is called as vibrio. Fusiform bacilli with tapered ends. Okay, so if they are bacilli but they have tape, see, you can see over here, you can see this tapered ends. My cursor, can you see over here?
Yes,
If
ma'am.
they have tapered ends, they are called as fusiform. And if यहाँ पे comma वाला shape नहीं है, but there were small commas, they are called as vibrios. Okay, here you will have commas. Yeah, this one is the comma one. Okay, vibrio. Can you see this?
Yes, ma'am.
Yeah. And then see, coccus may you have three. Okay, coccus, diplococcus, and what? Streptococcus. Too many over here. Clusters. Cocci in pairs are called diplococci, in chains are called as streptococci, and if it is in cluster, they are called as staphylococci. You should remember this. This you need to remember. You always tend to get confused. Okay, you remember this. If it is a chain-like structure, it is streptococci, and if it is in bunches like grapes, it is called as staphylococci. So the bacteria, if they are, when you see under the microscope also, it is easily viewable. You know, you can see single cocci like this. There'll be single dots like this. Or if it is diplococci, you can see two cells join. And if it is staphylococci, you can see a whole bunch together. When you see it in bunches under the microscope, when you observe it in bunches, you call it as staphylococci. And when you observe it like chains, you call it as streptococci. Okay. Are you all understanding?
Yes, ma'am.
Yeah. Okay.
Yes, ma'am.
Then this is vibrio. I told you comma-like are called as vibrio. Then bacilli mein there are two. One is bacilli, one is cocobacilli. Cocobacilli means there will be little bit of circular and little bit of elongated. Okay, cocci is complete circle. If you say cocobacilli, it will be a little bit elongated but round. Okay, so you said a cocobacilli. Then diplococcus will be two. Diplococobacilli, it will be two. Okay, and when at chain, there are streptobacilli. If it is in chain, it is streptobacilli. Okay, clear the slide, all of you?
Yes, ma'am.
Yeah. And this is how each type of cell and shape arrangement, how it looks. Okay, the cells, how do they look? This is cocci. This is bacilli. Okay, and then you're in chains over here, so it is streptococci. Okay, and these are comma-like. This is not, this is also streptococci. Okay, this is streptococci because they are in chains. And if you see staphylococci, there'll be bunches. I have another PPT, I'll show you. It has it will give you a much clear image of these bacteria. Okay, that will show you the streptococci under the microscope. Okay, these are bacilli. These are bacilli in chains. This is streptococci in chains, and bunches will be staphylococci. Clear all this to you all? They classify depending upon their cell wall. They classify depending upon their oxygen requirement, anaerobic, aerobic. Then they classify depending upon their shape. Cocci, bacilli, okay, streptococci, staphylococci, comma-like is vibrio. So these are depending upon their shape. So this was about the structure of a bacterial cell wall. Now we have the next topic, that is difference between prokaryotes and eukaryotes. You have a topic in your syllabus, second unit, you have differences between differentiate between prokaryotic and eukaryotic cell. That day I was asking you the difference between prokaryotic and eukaryotic cell. There are many points. You could tell me only a few, couple of points only you could tell me, but there are around 20 points of differentiation between the prokaryotic and the eukaryotic cell. Okay. If you all are sitting with a paper and pen, you can note down the difference also. Do you all have a paper and pen beside you?
Yes, ma'am.
Yes, ma'am.
Who said that? Who was that? Who said yes, ma'am, first? I think it was Muzammil and Gautam, right?
No, not me, ma'am.
Yes, ma'am.
No,
Me. Gautam, ma'am.
Gautam. So when you answer, you also tell your names, okay? So that I know who is having paper and
Yes,
pen.
ma'am.
Okay, it will be better if you note down so that you remember. You know what happens after a class? We just close our laptops or phones, and then we tend to forget whatever was taught. So if you note down, it is always better if you write it, you remember it always. Once written, you don't forget. Or next time, if you look into the paper also, you try to recollect what all was taught. Because after the class, we are out of the class, we forget whatever was taught in the class. Hmm? If you can note down the important points. Because that day when I asked you, you all said it's only nuclei बताया था मेरे को और क्या बताया था तुम लोग मेरे को? You all told me nucleus and you all told me only 2 or 3 points for differentiation. Write down all this at least. Cell size, nucleolus, chromosome, wherever it makes difference. See, now we'll discuss about this. Okay, prokaryotic cell and eukaryotic cell. Prokaryotic cell, under this you have bacteria and cyanobacteria, and under eukaryotic you have to study algae, fungi, protozoa, plant and animal cells. Okay, so let us see the differences. Cell size, what is it here? It is 1 to 10 micrometer, and eukaryotic, whereas cell size goes to 100 micrometer. Okay, note down if possible, you all. Then genetic material. Okay, here it is nucleoid chromatin body or nuclear material. Nuclear material is just present inside the cell, whereas here it is a defined nucleus, mitochondria and chloroplast. Okay, that is a eukaryotic. Structure of nucleus not bounded by nuclear membrane. Over here it is bounded by nuclear membrane. I will ask you all the points and then you have to tell me. Okay.
Okay, ma'am.
So structure of nucleus, there are almost 20 points. Try to remember if you don't want to write. Others, please note down. Structure of nucleus is not bounded by nuclear membrane and bounded by nuclear membrane. Nucleolus over here is absent and it is present in a eukaryotic cell. Okay. And then chromosomes. Coming to the chromosomes, how are they present? They are present in single or circular chromosome is present. Over here, it is linear chromosomes are present. Chromosomes contain histones, then mitotic division present, and nucleolus present. Functionally related genes are not clustered. Over here, what it is a single circular chromosome present. Chromosomes do not contain histone and no mitotic division. Okay. This is you, and you remember mitosis, meiosis.
Yes, ma'am.
Yes, ma'am.
This all of
Yes,
you have
ma'am.
done. So who will tell me the difference between mitotic and mitosis and meiosis? All of you have done this, mitosis and meiosis. Who will tell me the difference between mitosis and what is mitosis, what is meiosis?
Meiosis is
Genetic
reduction
material
division.
constant in mitotic division.
One of
Mitosis occurs in vegetative cells, ma'am, whereas meiosis occurs in reproductive cells.
who said that?
Gautam, ma'am.
Gautam. Who else is going to tell me? सबने पढ़ा है mitosis, meiosis. I'm not giving any marks over here. I just want to know how much you all know so that from there I can tell you. Anybody else?
Constant number of chromosomes in mitotic division, ma'am. From parent to
Meiosis is reduction division.
It was Deepak, I guess.
yes, ma'am.
What else? Who is going to tell?
2 daughter cells in mitosis. 4 daughter cells in meiotic division.
I couldn't hear. What did you say?
2 daughter cells produced in mitotic division. 4 daughter cells produced in meiotic division.
Who is saying that?
Deepak.
Deepak. Anybody else? Anybody else can tell me?
Mitosis, two daughter cells produce. In meiosis, four daughter cells.
Yeah, that just now he said. Anything else?
Single nuclear division in mitotic division, ma'am. Two nuclear division in meiotic division.
Deepak.
Yes, ma'am.
Who is telling like Akhila?
Mitosis, diploid cell division. Meiosis, haploid cell.
Anybody else?
Crossing over occurs in meiotic division. There is no crossing over in mitotic division.
Deepak.
Yes, ma'am.
I want someone else to answer. Akhila? Akhila is there. Harsha? Naveen?
Madam, meiosis occurs only in diploid cells, whereas mitosis can take place in both the type of cells, only diploid or haploid.
Who is telling that?
Naveen, madam.
Naveen is telling. Can you repeat what you said, Naveen?
Meiotic division takes place in diploidic type of cells, whereas mitotic can take place both in diploidic and haploidic type of.
What else?
Ma'am, mitotic daughter cells are genetically identical. In meiosis, daughter cells are genetically different.
Who said this?
Harsha, ma'am.
Harsha. So you all have so many points for mitosis and meiosis, huh? Everybody saying different version of mitosis and meiosis. Go, all of you, I suggest to go and revise mitosis and meiosis after the class. Muzammil, any idea?
Yes, ma'am. The same point.
Same points, huh? Same points. What did they tell? Can you tell me? What was told?
In mitosis, two daughter cells will be formed. In meiosis, four daughter cells will be formed. And meiosis is a reduction division. It takes place in gametic cells, and mitosis takes place in vegetative cells.
What else?
And meiosis specifically occurs in diploid cells, and whereas mitosis takes place both in haploid and diploid cells.
So याद है सबको mitosis meiosis?
Yes, ma'am. Chromosome numbers remain same in mitosis, ma'am. Reduced by half in meiosis.
So here we are saying it is no mitosis division in what we are studying prokaryotic cell. There is no mitotic division, but mitotic division is there in eukaryotic cells. So the number of cells are increasing or decreasing.
Increasing.
In prokaryotic, what will happen? Number of cells? No mitotic division, they're saying. Okay. So in prokaryotic cell, what will happen? You'll just now said mitotic.
Cell directly divides into two daughter cells.
Okay. Yeah. And nucleolus absent, nucleolus present. Okay. Functionally related genes are clustered. Functional related genes are not clustered. So basically remember these points, that cell size, then the genetic material, then structure of nucleus. If it is bound, not in prokaryotic, it's not bound. In eukaryotic, it is bounded to the nuclear membrane. And I'll ask all this in the next class. So remember all this. Genetic material, okay? It is nucleoid chromatin body or nuclear material, nucleolus, nucleus, mitochondria, and chloroplast. Okay, all these are the genetic material location. Then about the chromosomes, they are single circular chromosome and here there many linear chromosomes are present. Chromosomes do not contain histone, and here they contain histones. No mitotic division present. Okay, nucleolus absent, nucleolus present. Then coming to DNA. DNA, how it is? What about DNA in prokaryotes? And this is DNA in eukaryotes. So what happens in prokaryotes? DNA is located in chromatin material and in plasmid. You have seen the cell, no? I showed you the cell structure. So DNA is located in chromatin and plasmid, and whereas in eukaryotes, DNA is located in nucleus, chloroplast, and mitochondria. Okay, clear all of you? Here it
Yes,
is
ma'am.
single-stranded of molecule of
Yes, ma'am.
double-stranded circular DNA. Okay, and not complex with histones because यहाँ पे no histones do not contain histones, whereas these contain histones. So usually complexed with histones. Here it is a single molecule with double-stranded circular DNA. Here it is a double-stranded DNA molecule present in several chromosomes, complex with histones. Okay, remember the difference for DNA also. Division, no mitosis. Mitosis occurs. Mitotic apparatus with microtubular spindle is present. Remember this. Division is no mitosis, and here it is mitosis occurs. Okay? Mute yourself, whoever it is. Then reproduction, fragmentary process, no meiosis. Only portion of genetic material is usually asserted. And here, regular process, meiosis occurs, reassortment of whole chromosome, complement. Are you all able to hear me?
Yes, ma'am.
Yes, ma'am.
So, sexual reproduction, here it is fragmentary. And here it is a regular process, that is with meiosis. Okay. And in genetic recombination, unidirectional transfer of DNA by conjugation, transformation, and transduction. You'll have studied this conjugation, transformation, and transduction. Have you studied
Yes,
this?
ma'am.
Yes, ma'am.
Conjugation, transformation,
Yes,
and
ma'am.
transcription. Do you remember conjugation? Just now we said there is a formation of conjugation tube. What happens in transformation and transduction? Can you tell me? What happens in transformation and transduction? You all have done in 12th transformation and transduction. Just tell yes or no. I'm not giving any marks. I told you I'm just asking and talking.
Yes, ma'am.
Transformation,
Yes.
transduction. No, so you'll remember conjugation because of the conjugation tube. What about transduction and transformation? Do you remember?
In transaction, transfer of genetic material from one bacteria to another bacteria through bacteriophage.
In transformation,
Ma'am, in
Transformation.
transformation, the cell it takes the genetic material from outside environment and changes into that.
who said that?
Gautam, ma'am.
Gotham. I guess someone is sitting with a book also. I can hear them turning pages. Okay. Just we will be doing this in detail. Conjugation, transformation, transduction. We will be doing this in detail. You have it in your syllabus. Revise your thing. Whatever you all have studied, you all revise again. Because all that is there here again in your first sem and second sem syllabus. Then coming to zygote, nature is partially diploid, and here it is diploid. Cell wall present in most forms and mainly composed of peptidoglycan, and here in eukaryotes present in plants, algae, and fungi, and is composed of polysaccharides absent in animals and protozoa. Okay, so here the cell wall is composed of peptidoglycan, and here the cell wall is composed of polysaccharides. Okay. Then, do you all remember this? I'll go to the next slide. Can I go to the next slide? You all will remember
Yes.
this? See,
Yes, ma'am.
first what we had done the size. Then it was what? It was nucleus. Then it was what? What else was there in the previous slide? Who will tell?
Nucleolus.
Nucleolus. And what else was there? Previous slide.
Chromosomes.
Chromosomes. What else?
Next, DNA.
DNA तो यहाँ पे आया है. What was there in the previous slide? You have cell size, you have
It was
genetic
a
material,
chromosome.
nucleolus, chromosomes. Okay, remember this. Then you have DNA, you have division, you have sexual reproduction, you have genetic recombination, cell wall, and zygote. So all these points you have to write. You don't think that what madam is telling. You get a question, differentiate between prokaryotes and eukaryotes. Generally comes for a long answer. So you have to write all these points of differentiation when the question is differentiate between prokaryote and eukaryotic cell. To draw the structure of prokaryotic cell, you have to draw the structure of eukaryotic cell, and then you have to write all the differentiation points between prokaryotic and eukaryotic cell. Okay. Then next is the plasma membrane. I told you there are many points. Till now, how many are done? 10. Who is writing it? Nobody? Nobody is writing it?
12, ma'am.
12 are done till now. Who said that?
Navyasri, ma'am.
Navyashri. Okay, good. So, and there are many more over here. Okay. See, there are so many. So we'll go one by one. That's what I'm telling you. If you don't write, you'll not remember. Then coming to plasma membrane. It usually lacks sterols, and sterols are usually present. Sterols also you have studied. You have done sterols also. What are they? Fatty acids. Sterols. So in plasma membrane, they do not have sterol, but in eukaryotic cell, they have sterols. Then internal membranes, relatively simple and comprises of mesosomes. Here over here it is complex, includes endoplasmic reticulum, Golgi apparatus and all. We remember the structure of eukaryotic cell, no? All of you?
Yes, ma'am.
Yes,
You
ma'am.
have the endoplasmic reticulum, you have the Golgi body apparatus. Okay, all this is there in the eukaryotic cell, which is not there in the prokaryotic cell. So this is, we're talking about the internal membrane. Okay, then coming to endo and exocytosis, absent in prokaryotes, whereas present in eukaryotes. Then coming to ribosomes. Ribosomes here, we saw the picture of ribosomes in the slides previously. Ribosomes, I showed you the structure, no? You remember? You forgot?
Yes, ma'am.
Yeah. So ribosomes here are 70S, that is 50S plus 30S, and the two subunits. Okay, there are two subunits, 50S and 30S. Whereas in eukaryotes, it is an 80S ribosome. Okay, it is an 80S ribosome which subunits 60S and 40S, except for mitochondria and chloroplast. Okay, in those the ribosomes are 70S. Simple membranous organelles. Okay, in case of prokaryotes, they do not have any other organelles other than the plasmid and the genetic material DNA and all. They do not have any other organelles. But here you have the vacuoles, you have lysosomes, you have perioxysomes, mitochondria, ER, Golgi body, etc. Okay, so this is the eukaryotic cell which has other organelles, but whereas the prokaryotic cell doesn't have any other organelles. Okay. Then coming to the respiratory system. In prokaryotic cells, how do they respire? With the plasma membrane. They respire with the plasma membrane. Whereas in eukaryotic, you know, mitochondria. Mitochondria, what is the other name given to it?
Powerhouse of the cell.
Powerhouse, yes. So mitochondria are also called as the powerhouse of the eukaryotic cell. Okay, because of the respiratory system being present over here. Then coming to photosynthesis in the form of organized internal membranes or vesicles. Okay, chloroplasts are absent. So chloroplasts as such are not, other than blue-green algae, chloroplasts are not present in bacteria. Cyanobacteria have it. but here mostly they do not have, and other internal membranes, they work as photosynthetic apparatus. Okay, whereas here we know in a eukaryotic cell we have chloroplast. All of us have studied chloroplast in a eukaryotic cell. So photosynthetic apparatus over here is a chloroplast in case of eukaryotic cell. Okay, then the gas vesicles. Do you remember gas vesicles? How do they look? How do they look, gas vesicles?
Small air bubbles.
Yeah, and small vacuoles inside the cell. Okay, so they are present in some, whereas they're absent over here. And then endospores present in some forms and are very heat resistant. Absent in case of eukaryotic cell. Endospores just now we have done. We've done endospores just now. Okay, so they are present in some forms and are heat resistant, whereas it's not there in.
Not clear, ma'am.
Can you hear now? Can you hear now, Deepak?
Yes, ma'am.
Someone said they are not able to hear. Okay.
I think Manikanta, ma'am.
Manikanta, others are able to hear, no? I think some problem with his system.
Yes, ma'am.
Yeah, some problem with his system, I think. Okay. So endospores, we just noted endospores in the previous slide. It is present in some forms and are very heat resistant, whereas this is absent in case of eukaryotic cell. Okay. Then coming to flagellar movement. Flagella are submicroscopic in size. Each flagellum is composed of one fiber. Okay. Each flagellum means they're talking about a single flagellum. So each flagellum is composed of one fiber. Whereas in case of eukaryotes, flagella or cilia, microscopic in size, composed of microtubular elements arranged in a characteristic pattern of 9 outer doublets and 2 central singlets. Okay. So here in case of prokaryotes, it is submicroscopic, means you cannot even see it. You need the electron microscopes to see the flagella. Okay, but here it is not like that. In case of this eukaryotic cell, you can view it under the microscope. Okay, clear?
Yes,
So
ma'am.
till now,
Yes,
what
ma'am.
all points we have done? What are the points we have done till now? Who is going to tell? Which points we have done till now, starting from?
Flagella.
Starting se batao. Who is going to tell? First, what we have done is what we have done?
Groups.
Plasma membrane.
Groups. Then we have done what we have done?
Size of the cell.
Size. Then
Internal
Genetic material location.
yeah. Then
Structure. Nucleolus.
nucleolus present or absent. Then what we have done?
ribosomes.
Structure of
Ribosome.
nucleus.
Yeah. Then we have done
Chromosomes.
chromosomes. Then division.
DNA,
Division.
ma'am.
DNA. And what else?
Sexual
Cell
reproduction.
Structure of
Sexual reproduction, ma'am.
yeah, sexual reproduction. Then
Genetic recombination.
genetic recombination. And
Zygote.
who is answering?
Deepak, ma'am.
Others, can you tell me?
Cell size.
Cell wall.
Plasma membrane.
See, all of you should concentrate. It's not that only one person will concentrate and one person will answer. Right, when we started, I told you, remember these because they're almost 20 points. Remember this, someone was writing it also. Cell wall, there is a difference in cell wall also. Cell wall में क्या difference है? Peptidoglycan and here it is polysaccharides. Okay, peptidoglycan in case of prokaryotes and polysaccharides in case of eukaryotes. Okay, then we have done plasma membrane, internal membranes. Then we have done ribosomes. Then we have done simple membranous organelles. Then respiratory system. then gas vesicles, endospores, and flagella movement. Okay. Coming to this next points. Will you remember? Are you all remembering? What I'm telling you, try to recollect or write down. That's why I told you if you forget, try to write down. Okay. So next is, that was flagellar. We were studying, we said about flagellar movement, and then this is non-flagellar movement. So some cells which do not have flagella. Okay. In case of prokaryotes also, some cells which do not have flagella, they have gliding motility, gas vesicles mediated motility. So here in this case, what happens? They have gliding motility. If they do not have flagella, they have gliding motility and gas vesicle mediated movement. Whereas here, in case of eukaryotic, if the cells do not have flagella, it is cytoplasmic streaming, amoeboid movement, and gliding motility. Amoeba also you all have studied. Remember amoeba, no?
Yes, ma'am.
Yes,
Yes,
Yeah.
ma'am.
So they do not have flagella. They do not have the flagella appendages, but they have this amoeba. They have pseudopodias, and they move. That they also amoeboid movement. You know what is amoeboid movement, no? They move with the help of. That is also one of the gliding movements. Okay. Then appendages here are pili and flagella in case of prokaryotes, whereas flagella with whip-like motion. They have a big flagella, no? If it's eukaryotic cell, they have a big flagella. Then cytoplasmic, streaming, absent and present. Okay. Locomotive organelles, simple fibril, and here it is 9+2 configuration, means it will have 9 and 2. Pseudopodia. Pseudopodia, prokaryotic cells do not have pseudopodia. Yes. But in eukaryotes, pseudopodia is there. DNA base ratios is as mole percentage in guanine and cytosine. Okay, percentage of guanine and cytosine 28 to 73, and here it is about 40. Okay, mitochondria in case of prokaryotic cells it is absent, whereas in case of eukaryotic cells it is present. Chloroplast also absent in case of prokaryotic cells, but present in case of eukaryotic cells. Then Golgi structures. Do they have Golgi structures? Prokaryotes?
eukaryotes
Yeah, prokaryotes do not have the Golgi structure. And endoplasmic reticulum?
present in eukaryotes.
Yeah, prokaryotes do not have the endoplasmic reticulum also. Okay, so you remember this slide? You remember
Yes,
this?
ma'am.
Yeah, in the previous thing, what we did, ribosomes, membrane, respiratory organs, photosynthesis apparatus. So it is not one or two, it is 20 and more, if anybody has written. There are many points of differentiation between prokaryotes and eukaryotes. I hope you all understood this. Mitochondria absent in prokaryotes, chloroplast absent, Golgi structures absent, even endoplasmic reticulum absent. Whereas all this present in case of eukaryotes. Clear this thing?
Yes, ma'am.
Clear all of
Yes,
you?
ma'am.
Tomorrow I will ask you, you should tell me in the next class what we have done today. I always have a habit of revising the previous class. Okay. So whenever I take a class, I'll be asking you what was done in the previous class, and I expect your answers. I expect you all to tell me what was taken in the previous class. Okay. Yeah. Did you all understand what we have done today? Structure of cell, cell wall, and difference between prokaryotes and eukaryotes. Complete lot of differentiation points I've told you all. I hope you'll remember and you'll recollect tomorrow. Can you switch on your videos? Can you switch on your videos for a screenshot? Okay. Do you remember, Nikita, did you understand today what all was taken?
Yes, ma'am.
Okay. Who else is here? Hafsa, was it okay today?
Yes, ma'am.
Okay. Then Navyashri. Navyasai.
Navyashree, ma'am.
Navyashri. Yeah, sorry if I spell your names wrong, but
Okay.
I'll know your name soon. Okay. So, and who is this? Who else is there in the class? 1 minute. Okay. Anjali, was it okay?
Yes, ma'am.
Then, if you don't understand anything, you can ask me. Okay. And this is Shri Harsha.
Yes, ma'am.
Was it okay? Naveshri?
Yes, ma'am.
Yeah. Okay. Are you all understanding whatever I take? Okay. Okay. Any doubts, you can ask me. Okay. See you all in the next class tomorrow. Right? Bye.
Bye, ma'am.
Bye bye.
Bye, ma'am.
Bye, ma'am.